Hello students, welcome back. Today I am going to discuss the structure of antibody. In last lecture I have given you the theoretical part of the antibody structure, but today I will show you a diagram and we will understand the structure of antibody and why it is called H2L2. So you can see the diagram. The antibody structure is Y shape. You can see that on the topmost part of the structure it is labeled as antigen binding site. Now what does it mean? It means that any foreign material or antigen, when it enters the body, this antibody it's reached to that and the antigen gets trapped in that Y shape, or you can say that binding site. Now that binding site is variable part, so it has different shapes according to the different antigen. They are complementary, so the antigen can be easily trapped or caught by the site. Another important feature of this antigen binding site is it has N-terminal. Now what is N-terminal? If you see amino acid structure, a polypeptide structure, basically immunoglobulin means polypeptide and polypeptide means number of amino acids, right? So amino acid structure, if you see, on one side it is NH3 and the other side it is COOH. So we can say on one side it is N-terminal and the other side is C-terminal. The same thing applies here. The N-terminals is on the antigen binding side and the C-terminal is on the base of the Y shape. Now if you see the diagram, you can find that in this antibody structure, the Y shape is joined by disulfide bond. You can see SS bond within a chain and between two chains. So you can see number of SS bonds, disulfide bonds. You can also see the diagram very carefully that there are two long chains and there are two short chains. The long chains are called heavy chains, the short chains are called light chains, and therefore it is called H2L2 because there are two heavy and two light chains. And as I already discussed that IgG is the most prevalent antibody in the blood, followed by IgA and IgM. IgA is present in the colostrum, that is a breast milk also. Now you can see that the structure of antibody, okay, we have seen immunoglobulin 5 types: IgG, IgA, IgM, IgD and IgE. All these antibodies, Y-shaped immunoglobulins, has variability in their antigen binding site. So if there is a question that write a note on antibody, so what you can do, you can draw this, you can label it, and you can write about the antibody. The question may be of 2 marks or 3 marks, and if it is 3 marks, compulsory you have to draw the diagram, label the diagram, okay, mention all the things which I have already discussed. Draw a Y-shaped molecule which is shown in the diagram, label it N-terminal and carbon terminal, C-terminal, then antigen binding site, disulfide bonds between the chains and within the chains, heavy chains, light chains, H2L2, IgG most prevalent, IgG, IgA, IgM, and antibodies are also called immunoglobulins. Basically they are proteins made up of polypeptide chains. So at least this much you should write and you can draw. You can easily get 2 or 3 marks depending on the question. Fine. So this is the structure of antibody. I hope it is clear. This structure and the content which we have discussed is limited to class 12th NCERT only. There are detailed structures and detailed theoretical part, but we are not going in detail because we are restricting ourselves with class 12th NCERT book only. So if you have any doubt, you can comment me, you can ask me on my WhatsApp number also. Right? Thank you.